So all you have to really look at when you're looking at mitochondrial DNA is you have to worry about point mutations and sometimes insertions and deletions, but you don't have to worry about this gene shuffling that happens in the nucleus. And that just makes things cheap and easy to make these evolutionary trees using mitochondria. The next slide. Mitochondria has a small genome in comparison to the rest of our genome. It's only got 37 genes. Your nuclear genome has about 20,000. And then the mitochondrial genome has 16,569 nucleotides in it in humans. It varies a little bit in different organisms. One mitochondria usually has several copies. So you can see I've got several little loops of DNA inside this mitochondrion. But their genome's simple. And that means it's easy, quick. We can sequence it relatively cheap. Next slide. The fascinating thing about mitochondria, and this is kind of an aside to the book because Jeanson doesn't talk about this at all in there, but the fascinating thing about mitochondria is where the heck they come from. Their DNA works different than our DNA does. As we saw, there's this round thing, this round chromosome instead of the X chromosomes that we have in ours. We have found out recently that they are bacteria. Mitochondria are organisms that live inside of our cells. And the merger between cells that eventually gave rise to us and bacteria, we think from the fossil record, it's a little bit blurry, but it seems from the fossil record that this happened 2 billion years ago. There was a proto-eukaryote. So we are eukaryotes. We are organisms that have nuclei inside of our cells, and we have mitochondria. So there was a proto-eukaryote, which may or may not have had a nucleus before this interaction. A proto-eukaryote, and somehow a bacteria got into it, got inside of its body, and started reproducing and living in there. And the two organisms evolved symbiosis. And this is absolutely fascinating because almost all eukaryotes now, it's not true for all of them, but almost all eukaryotes, if you kill their mitochondria, the cell that those mitochondria live inside, that cell will die too. We are now fully dependent on our mitochondria. That obviously wasn't the case before this merger, but since we've been living together, it's kind of like a marriage, like you used to be totally independent and do all your own stuff, and all of a sudden you don't know your bank account because you've been married so long, and that's what your partner takes care of, So this same sort of thing happens over evolutionary time. When you have two organisms that come together in symbiosis, they actually lose the ability to survive on their own, and they start dividing chores and kind of dividing and conquering, separating their tasks. And this is what's happened in eukaryotes. Almost all eukaryotes, we are prisoner to our mitochondria, and our mitochondria are prisoner to us. And I'm not going to go into it here, but there is some surprisingly strong evidence that the reason that we have sex, the reason we reproduce sexually is because of mitochondria. The reason that we are able to be multicellular is because of mitochondria. And the reason that we have male and female, so the sex is because of mitochondria. If it weren't for them, if it weren't for this event that happened, it was a single event that happened, we think 2 billion years ago. If it weren't for this event happening, none of that stuff would have happened. It's just absolutely crazy. These things
that's
are so
recently?
important.
2 billion years ago is recently?
No, we recently discovered that this is what happened.
Gotta listen, Steve.
That relationship you were talking about, where you have mitochondrial, the DNA it's like a marriage, and one can't survive without the other. That's kind of the relationship that Steve and I have now, because he definitely wouldn't be able to.
He's the bacteria.
Kyle's the one that made the whole thing sexual. I guarantee it.
That's fair. That would be fair.
That would be an accurate representation, yes.
Yeah, and by the way, you're right. You did say, I will correct it. You did say that we discovered it recently, which was back in the '60s. Was it Margulis? That
yeah, Lynn Margulis. Yep. She's the one that got everybody on board with it. It was the discoveries were made before by one guy who's very people don't like to mention him very much because he was a horrible dude, but she's the one who really got it publicized and got it accepted in the scientific community, and she added a lot to it as well, making sure that we would notice this. And it's so obvious, it's crazy we didn't notice it before, but it's a very significant discovery and good work.
There's one more question. There's one more question that asks: do other organelles in the cell have a symbiotic relationship with its host?
Yes, chloroplasts do, and there are several others that might, but we can no longer tell for sure.
Chloroplasts are only plant cells, though, aren't they?
Yeah, some animals get them too. Like some corals will have.
They're only
corals
for
will have
photosynthesis,
symbiotic.
generally, aren't they?
What's that?
Are they only for? I thought that they were only for photosynthesis. I could be wrong.
Chloroplasts do photosynthesis, and they excrete sugars into the host animal. Yeah, or the host.
They help
plant.
with that metabolism within the cell. But
how they came about was a very similar thing, right? The chloroplast, they came about by a symbiotic type relationship as well, right? Is that what you're saying?
And we have, I think there's lots of instances of this that are not symbiotic, they're parasitic. So we have, I think it's chlamydia. I'm pretty sure it's chlamydia, or it's
I've never had a symbiotic relationship with chlamydia. I'll tell you that right here now.
Always goes to chlamydia. It doesn't matter how serious it starts. Never mind.
It's a bacteria. If I'm remembering this right, it's one of the STDs that's like going around college towns everywhere. But it gets into the cells of the urethra and starts breeding inside of there. And I believe it ruptures and goes into other cells afterwards. So it's parasitic. But most symbiotic relationships start out parasitic. So we could, 10,000 years in the future, like chlamydia is like metabolizing your whatever for you. That could be a thing in humans of the future. Because
so we all have little pet chlamydias maybe or something, You go buy a little chlamydia at your local Petco or something. Chlamydia.
We have so many organisms that live in us that help us, in our digestive system, and then we even have mitochondria in our cells. It's amazing. It's a really fascinating kind of new way to think about some of these big evolutionary changes happened not through mutations but through merging of two organisms. Really cool. Anyway, next slide here. That was all in the book.
Yeah, after this transition, let's see what happens now.
Yeah. Anyway, eukaryotes are organisms that have sex and that have mitochondria. There are a couple of exceptions, but in every exception that we've studied so far, it's evident that the organism used to be able to have sex and used to have mitochondria. So even these single-celled organisms, these protists, they do reproduce. They'll reproduce asexually sometimes just by splitting, but they also reproduce by making copies of their DNA and shuffling those copies with a partner. So there is sexual reproduction, and plants do this as well with pollen, and the seed has to get fertilized by pollen. That's sex is the shuffling of genes to diversify the genomes of your offspring, and this is all eukaryotes either do this or used to do it in the past, and all of them either have mitochondria or used to have mitochondria in the past. And next slide. Here's an actual image of mitochondria. They actually come in all kinds of different sizes. These ones are round, but they can even fuse with each other. If one of them's acting funny, sometimes they'll fuse together, and they'll donate DNA to each other, and their DNA they'll pair up and try and repair each other. It's really cool. But sometimes they're cylinder shaped, sometimes they're round like this. These things caused all the cool stuff to happen in life. They're responsible for making you fall in love. They're responsible for your divorce. All kinds of cool stuff. So next slide.
That is fascinating to think about, I think. It's crazy how that all works.
Yeah. These, I said some of these organisms have lost their mitochondria. This is Giardia. And Giardia is the disease you get if you drink, it's the things that get inside your intestines if you drink dirty water, like pond water. I think this was a gerbil or a hamster they got this from. Those things are covering its intestinal wall. It can't get hardly any food. So it's eating, doesn't get hardly any of that. They're getting it all. And they're just covering this poor animal's intestines. These things have lost their mitochondria almost completely. They have found a way to produce their own ATP. I didn't even mention that, but the big thing that mitochondria do for us is they produce ATP, which, we think of food as fuel, like we power our bodies by eating food. Well, it's not true. We actually, our cells actually only use a specific type of fuel. They use ATP, and mitochondria help convert our food into ATP for us. So mitochondria are kind of like an oil. It's kind of like an oil refinery that converts crude oil into gasoline for your car.
Everybody in the live chat would like to know a little bit more about ATP synthase, by the way.
Oh, please. Oh, goodness.
Oh, no. There's a running joke on this show about ATP synthase, because we had a gentleman who was arguing for intelligent design say it on one of our shows about 96 times, I think somebody counted. But that was his evidence.
That's how bad it was. Somebody literally went through it and counted how many times they said it.
Yeah. That was their evidence for why we're intelligently designed. So it's ATP is infamous here.
Yeah.
Infamous.
Yeah. Well, he should study Giardia, because Giardia figured out how to do it on its own again throughout evolutionary time. A really fascinating animal. Not an animal. It's a protist. Anyway, next slide. Oh, here's just that tree again. I don't know why I put that in again, but it is cool to look at.
It's a cool tree.
So his tree, by the way, he used mitochondria to make this. He didn't use hemoglobin. He used mitochondria. So the lengths of those branches show you how distantly related those animals are by differences in mitochondrial DNA. Next slide. So the pros of using mitochondrial DNA as a clock is that it's really high resolution. I didn't really talk about this, but it evolves faster than the rest of our genome does. And there are several reasons for that, which Mays might talk about, because it's kind of important for some of the arguments they might have. It does evolve faster. More importantly, mutations in mitochondria reach fixation faster. There is no genetic crossover, like I mentioned earlier, in mitochondrial DNA. It's easy to sequence for us. Now that we have these machines that can sequence DNA, it's fairly cheap and easy to do. And then almost all eukaryotes still have mitochondria, meaning that we can build trees that include most plants and animals and fungi and protists. So we can make really nice trees with mitochondrial DNA. And that's certainly not true with a lot of the genes that we have in the rest of our genome. There's some genes that are just unique to mammals, for example. So it's a really nice thing to use when you're making these evolutionary trees. Next slide, we'll look at the cons here. The mutation rate varies in mitochondria according to how long a species' generation time is. Because, as you might recall, women, all of their eggs are already in them when they're fetuses. And all of those eggs go dormant. And what happens when they go dormant is their mitochondria essentially go to sleep. They just stop doing stuff. And the reason for that is that mutations in mitochondria are usually bad. And by causing the egg to go to sleep until the female is ready to reproduce, that makes it so that there's no mutations that accumulate in that during your lifetime. So it cuts down on mutations in mitochondria. Because of that, it screws up our evolutionary clocks, because mice reproduce a lot faster than humans do, right? And that's one of the reasons why we have that jagged edge on these mitochondrial clocks, because there's differences in generation time between different organisms.